everyone. My name is Yu Chen. I'm a cancer researcher from University of Newcastle. It's my great pleasure to be here to present my research funded by Cure Cancer. My research title is Detecting and Destroying the Hidden Devils for Bowel Cancer Treatment. So today I'm going to introduce what these hidden devils are, how we find them out, and our plans to detect and destroy them. Firstly, I'd like to have a brief summary for my journey towards cancer research. My experience might a bit different from other researchers because I was initially trained to be a doctor. I started to be interested in cancer research when I was a medical student, especially during my internship in the hospital. I'm very curious about what makes the uncontrolled growth of cancer cells and how we are going to use these biological discoveries to treat cancer. So to better understand this, I came to Australia and started my PhD degree focusing on cancer cell biology. In 2021, I obtained my PhD degree, and very luckily, at the same year, I was awarded my first fellowship from Cancer Institute New South Wales upon my PhD completion. And my fellowship was focusing on translational cancer research. My research have been mainly focused on bowel cancer. Australia has one of the highest rate of bowel cancer in the world. Each year, around 15,600 Australians are diagnosed with bowel cancer, and 1 in 3 of them will eventually die from the disease, making it the second deadliest cancer in the country. So why the prognosis of bowel cancer so poor? Actually, if bowel cancer can be detected at the early stage, it can be successfully cured by surgery. However, only less than 40% of bowel cancer cases can be successfully detected at early stages. For patients with late-stage bowel cancer, they are often treated with anti-cancer drugs, but most of them will eventually die from the disease because bowel cancer cells are resistant to the drug treatment, leading to the recurrence of the disease. So these findings highly suggest the urgent need for the new tool for early detection of bowel cancer and the new treatments for the late-stage bowel cancer patients. Currently, almost all currently used cancer biomarkers and drugs target proteins. This is not surprisingly because it was formerly believed the proteins are the only functional molecule that can regulate various biological processes to maintain cell growth. And indeed, a number of proteins have been reported to be aberrantly expressed in cancer cells, contribute to their uncontrolled growth. However, do you know proteins only represent 2% of human genome, leaving the vast majority of non-protein molecules underappreciated? So what are these non-protein molecules? They are called non-coding RNAs. Non-coding RNAs was initially thought to be junk or the dark matter of the genome because people believe they are not functional. However, we and others have established that long non-coding RNAs, which is a major subtype of non-coding RNAs, are frequently expressed at a higher level in cancer cells and contribute to their growth and survival. So based on these findings, we hypothesize if targeting long non-coding RNAs represent a novel avenue for cancer diagnosis and treatment. So inspired by this, we systematically analyze long non-coding RNA expression in thousands of cancer tissues compared to its adjacent normal tissues across 20 cancer types, including 600 bowel cancer samples compared to its paired normal adjacent tissues. And this has identified a lot of long non-coding RNA, which are commonly upregulated in different cancer types. Here we can see each dot represents a long non-coding RNA that is upregulated in one specific cancer type. So among them, we also identified a group of long-non-coding RNA that are specifically upregulated in bone cancer, including MILIP, PLAN, and REG1-CP, which we have already reported for their importance to promote bone cancer cell proliferation, growth, and survival. So further analysis of this long-non-coding RNA surprised us because we found their expression actually started increasing from the early stage during the bone cancer development, indicating they are the early event for the development of bone cancer. Moreover, we are able to detect them. We can stain long-non-coding RNA. Here we can see the black dot are the stained long-non-coding RNA, which can be seen in the bone cancer tissues, but not the paired normal tissues. These findings suggest long-non-coding RNA may represent potential biomarkers for the early diagnosis of bone cancer. So given the high expression of long-non-coding RNA, we ask, what are their functions? So to better understand this question, we engineer our bone cancer cell lines so that we could be able to manipulate their expression. We inject the engineered bone cancer cells into mice and monitor the tumor growth. Very significantly, we can see in the group with low expression of long-non-coding RNA significantly inhibit the bone cancer growth, indicating the importance of the high expression of level of long-non-coding RNA in tumor growth. Let's have a look at the cells under the microscope. When we engineer the bone cancer cell lines, we additionally introduce green fluorescence into it to better visualization. So here we can see in the group with a high expression level of long-non-coding RNA showing this shining green fluorescence, which are significantly reduced in the group with low expression level of long-non-coding RNA, indicating long-non-coding RNA can promote the bone cancer cell growth. You may ask, okay, this is engineered cells, which may not entirely represent the original bone cancer cell growth. So to answer these questions, we use additional methods to directly reduce the long-non-coding RNA expression in original bone cancer cells. And here we can see similar to our engineered bone cancer cell lines, reduced expression of long-non-coding RNA significantly reduced the bone cancer number. So this finding highlights the potential of long-non-coding RNA as potential therapeutic targets for bone cancer treatment. So based on these findings, we have two aims. We'd like to test whether long-non-coding RNA can be targeted for bone cancer treatment and determine whether measurement of long-non-coding RNA expression can be used for early diagnosis of bone cancer. For aim 1, we are developing long-non-coding RNA inhibitors using nanotechnology. The mechanical basis of this study is a Nobel Prize winning discovery called siRNA-mediated gene silencing. So siRNAs are small, short nucleotide sequence that can specifically target one unique long-non-coding RNA and leading to its degradation. However, we are not be able to directly apply siRNA into our human bodies because siRNAs are very unstable and they are easily to be cleaned up by our circulation system. So what we need is a protector for them. So, and this is exactly what we do. So we use biosafe materials to design lipid nanoparticle that is able to encapsulate the siRNA inside to protect it and deliver it. The beauty of this design is, just as I mentioned before, the siRNA is unique. They specifically target one unique long-non-coding RNA. However, the lipid nanoparticle formulation can capture different siRNAs. So this is a one-size-fits-all design. We have tested the effect of this drug in our bone cancer cell lines. Here we can see treatment of this drug can indeed successfully reduce long-non-coding RNA expression level, leading to the reduce colony formation of bone cancer cells. And next, we are going to test the effect of these drugs in our bone cancer mouse model, which closely resemble the bone cancer growing in human bodies, potentially develop new drugs for bone cancer treatment. For AIM2, we are going to test lanthanum expression in blood samples and tissue samples to identify novel lanthanum biomarkers. We are also establishing collaborations with biopharma companies to aiming to develop a low-cost, easy-to-use kit for the early detection of bone cancer using these lanthanum biomarkers. This project will result in the new lanthanum biomarkers for early diagnosis of bone cancer and new lanthanum targeting the drugs for late-stage bone cancer treatment. The result will be progressed towards the commercial developmental stage to manufacture biomarker test kit and pave the way for the clinical evaluation of these new drugs in patients. We fully anticipate our research outputs will lead to better patient outcome and improve their quality of life, thus generating the benefits for the whole community. At last, I'd like to thank our team members, including our consumer representatives, our international and national collaborators, and our funding bodies, especially Cure Cancer. Thank you very much. I'm happy to take any questions.